Thank you for the introduction. I would like to tell you a bit about how we achieve enhanced productivity by modifying the Pichia secretory pathway. Just a quick general introduction of Validogen. We are a CRO based in the Graz area of Austria, Central Europe. Among our services are expression feasibility studies, development of production strains and bioreactor cultivations, as well as protein purification, furthermore some enzyme engineering as well, and cell banking. I won't go into any details here for time reasons. If you'd like to know more about our services, there are 3 of us here, so please contact us. We will be happy to engage in a conversation. We do all these services by applying parts of our proprietary Unlock Pichia toolbox. It consists, among others, of our exclusive AX1 promoter library for methanol-induced and methanol-free processes, various platform strains, plasmid system, novel helper vector, secretion signals, and various protocols. The promoter library consists of several types of AX1 promoter variants. There's the conventional type that features weak derepression and strong induction. Then the altered pattern with strong derepression and strong induction. Third type with strong derepression but weak induction. And finally type D, that's basically not derepressed at all and is strongly induced by methanol. Those variants that are interesting for methanol-free production are, of course, the ones that are strongly derepressed, and the ones for the methanol-induced processes are the ones that are strongly induced, of course. Now to the novel secretion signals. The secretion signal enables the protein to be transported into the ER lumen, which is the first step, the entry point to the secretory pathway. The general textbook knowledge is that there are two translocation modes, the post-translational one and the co-translational one. The gold standard, the most widely used secretion signal for Pichia is the alpha-mating factor pre-pro secretion leader from Saccharomyces cerevisiae, and it features the post-translational translocation. Briefly, in this case, the mRNA ribosome complex is built in the cytosol. The whole polypeptide chain is synthesized and transported through a pore into the ER lumen. With the co-translational mode, the mRNA ribosome complex docks to the ER membrane, and the growing polypeptide chain is transported into the ER lumen as it is synthesized. There are several potential issues with post-translational translocation, such as the protein could be degraded directly in the cytosol. It can trigger some cellular alarm signals. It can block the ER4, and it can also be toxic to the cell if it's already active in the cytosol. In theory, these issues are circumvented by applying the co-translational translocation. We tested 2 novel Pichia native signal peptides that were described as potentially co-translationally acting. We tested them both as standalone signals and also in fusion with the pro region of the alpha-mating factor. We used the GFP for first test as a model protein, since it very rapidly folds in a cytosol, and is therefore not very well secreted when the full alpha-emitting factor is used. We transformed two MAT-S Pichia platform strains without and with overexpression of PDI, protein disulfide isomerase, which is an ER-located folding chaperone. We screened 46 clones per each construct, and here we see the results on the left for the MAT-S strain and on the right for the MAT-S PDI strain. Now, for the basic MAT-S strain, there was basically no difference between the two novel signal peptides. We call them A and B for now, and the alpha-emitting factor. With the PDI overexpressing strain, there's a clear improvement over the whole group of the clones with signal peptide A, which is a clear indication that this signal peptide works in a co-translational mode. For the scale-up, we used the microbioreactor system BioLector Pro that still offers pretty high throughput, but here we can apply conditions that are very similar to those in a bioreactor, since we can measure and control online several parameters such as biomass, pH, dissolved oxygen, temperature, and so on. And most importantly, in this case, we can measure online the GFP fluorescence signal. And here we see the fluorescence signal of the best clones of the constructs with the alpha-emitting factor and signal peptide A. The black line were the empty control strains, so MAT-S and MAT-S PDI. The blue lines are the strains with the alpha-emitting factor. Red is the MAT-S strain without PDA and signal peptide A. And green, which clearly outperforms all the other constructs, was the PDI overexpressing MAT-S strain with GFP and the novel secretion signal A. So these results confirmed the microscale outcome. And we also tested the hybrid secretion signals with signal peptide A or B fused to the pro region of the alpha-emitting factor. And interestingly, these were overall the best performing clones. So at least for the signal peptide A, less so for the signal peptide B. This is the cell density at the end of the fermentation in the bioreactor, just to show that all the strains grew uniformly, so there were no growth effects. Okay, secreting GFP is fine, but what's most important is how do these signal peptides perform for secretion of other proteins, so of therapeutic or industrial relevance. To skip a couple of scales, these are results from bioreactor cultivations after applying same screening approach and size for all constructs. Here, only results from the hybrid signal sequences compared to the alpha-emitting factor are shown because the standalone signal peptides A and B were basically never better than the alpha-emitting factor. With these two therapeutic proteins, there was a 5-fold increase with signal peptide A over the alpha-emitting factor and 3-fold with signal peptide B. Even better for this industrial enzyme, 10-fold enhancement over the alpha-emitting factor and about 6-fold with signal peptide B. In some cases, the full-length alpha-mating factor doesn't really work at all. So here there was, so with alpha-mating factor, we only got a couple of milligrams per liter of these two proteins, and there was a 50-fold increase with the new hybrid sequences. So there are not many such cases, but occasionally alpha-mating factor just doesn't do the job. Then we went on searching for further signal peptides. After a genome-wide search, we did an in silico prediction of the cleavage probability and tested the further signal peptides firstly as standalone, and then again in the fusion with the alpha-mating factor. We narrowed the candidate list, so based on, of course, secretion efficiency of the model proteins, and also on the cleavage upon the entry into the ER, or actually, so if the signal peptide was cleaved from the final product or not, and those where the peptide was still present in the supernatant fused to the protein, so we ruled those out, of course. The first step again was GFP. Methanol, in the methanol-induced process, we found several peptides that clearly outperformed the alpha-mating factor, so all the results are normalized to the alpha-mating factor. This is the first column. For example, single peptide D with almost 5-fold improvement, or H or F 3 to 3.5. It was even better in the methanol-free process, up to 9-fold improvement over the alpha-mating factor with, in this case, single peptide C, E, or H. Single peptide G did not show any improvement over the alpha-mating factor pre-pro at all. Okay, we saw this graph already. That was the enabling, basically, with single peptides A and B for this industrial enzyme with a 50-fold improvement. And in the next test, the single peptide A was the benchmark. So this is the same construct, the same strain. And some of the novel constructs, like DE or G in this case, even outperformed the already excellent result from the signal peptide A. This is the same signal peptide G that was even worse than alpha-mating factor with GFP. Similar outcome for a therapeutic protein here called number 1. We used the signal peptide A construct as a benchmark to test the other group of signal peptides, and again got up to 2-fold improvement over the signal peptide A that was 5-fold better than the alpha-mating factor in the previous test. And now to several more case studies. So these are all, again, results normalized to the alpha-mating factor. Final titers were compared from bioreactor cultivation, always the hybrid constructs. So single peptide plus the pro-region of the alpha-mating factor. The green columns are from the methanol-induced process, and the blue ones are methanol-free. So the results were all over the place. So some were better under one condition, some are worse, and there were really large differences. For example, single peptide E for the single domain antibody outperformed the alpha-mating factor by a factor of 4 under both conditions. Single peptide C as well, 3-fold, but for example, single peptide F was clearly better when methanol wasn't used was used for induction, but was much worse than alpha-emitting factor in a methanol-free process. Similarly, for a single chain fragment, for example, single peptides A and B were much better in a methanol-free process than when induced by methanol. Still an improvement of about 50%, but not as good as, for example, single peptide F, 2-point-fold. And final case studies with tetravalent VHH and another industrial enzyme. Here again, so some of the novel signal peptides were better under both conditions, but signal peptide G, for example, stands out. Threefold improvement methanol-free, and basically the same result, or even underperformed in comparison to the alpha-emitting factor when induced with methanol. And with a final industrial enzyme, signal peptide C stands out as the best one under methanol-free conditions with a threefold increase, but it wasn't as good as the alpha-emitting factor under methanol. Whereas signal peptide G, for example, was 250% better under both conditions. So as many other things, it's protein dependent. To sum up, it's very important, of course, which secretion signal one takes for the expression of the protein of interest, and the alpha-emitting factor, the standard choice, is sometimes simply not a viable option. It's irrespective of the path, of the mode of translocation. So if it's post-translational or putatively post-translational, it's always worth testing different options. So it's as we saw sometimes, one signal peptide clearly outperforms the alpha-emitting factor, but then for another protein, it's just not as good. So we developed a panel of novel signal peptides are hybrids, so always the signal first and then the pro-region of the alpha-emitting factor second, that often outperformed the full-length alpha-emitting factor with many differences in secretion efficiency, so depending on the target protein, but also on the cultivation conditions, so whether it was a methanol induction or methanol-free processes. Finally, I would like to acknowledge our partner, Boehringer Ingelheim, with whom we have a non-exclusive joint technology development dating back from the old times when we were called V2 technology. Some of the results that we presented here were generated within this cooperation. Thank you very much for your attention, and I'll be happy to answer any questions.
Yeah, you showed that the effects of the co-translational translocation was really only seen in the PDI overexpression strains. Was that because it seems that PDI was a downstream bottleneck that was independent of the co-translation, or do you think it was actually directly involved in that effect?
Well, the way I interpret it is the PDI is necessary for effective folding. So the bottleneck was the transport of the polypeptide chain into the ER, where the PDI was able to help. When GFP folds fully in the cytosol, it probably blocks the pore, and it doesn't enter the ER as effectively as it does with the co-translational mechanism.
Thank you.
Thanks, Ait. This was fantastic. So I will have a question about the GFP secretion as well. It was shown that also much of the GFP is ending up in the vacuole, right? Did you check whether there is still a lot of GFP when you use the new hybrids of the signal sequences?
Not that exactly. So we just measured the fluorescence in the supernatant and in the whole broth, and it was basically the same. So we made we concluded that it's that everything that's correctly folded, is in the supernatant. We didn't do any fluorescence microscopy and so on. So we were just interested in how well it's secreted.
Okay, thanks.
Hi, great job. When you manage to overexpress better your protein thanks to the hybrid, do you get more hyperglycosylated forms, or is just the native form that you manage to overexpress more?
Sorry, I didn't quite get that. Do you get what?
So you manage to, for like the peptide A, C, G, to obtain more protein, right, versus the alpha-emitting factor. But you obtain more of the native protein, or you get more glycosylated form of the protein, right? Because Pichia have the tendency to hyperglycosylate when you start to overexpress it.
Yeah, it depends on the protein. So we didn't see any difference in glycosylation pattern when we used the alpha-emitting factor pre-pro and the novel hybrids. So if the protein is glycosylated by Pichia, it's glycosylated anyway, but in the same manner.